This video covers the T-cell dependent B-cell response for activation. This is part of the adaptive immunity process. When the B-cell matures in the lymphoid follicles, it can either be activated immediately by antigens called T-cell independent activation, or it can recirculate in the vascular system. This second type of B-cell has a limited life expectancy if it does not become activated. These B-cells require help from the T-cell to become activated. This is the T-cell dependent activation. This process starts when the B-cell binds an antigen on its receptor. This antigen is engulfed by the cell and broken down into peptides that can then be expressed by the MHC2 molecules on the B-cell. A wandering helper T-cell will pick up the signal from the MHC2's peptide complex on the B-cell. The cells require extra help from the CD40 molecule on the B-cell and the CD40 ligand on the T-cell to stabilize the connection. This binding will cause the proliferation and the differentiation of the B-cell and save it from apoptosis. Once the binding occurs, the helper T-cell will release cytokines such as IL-2, IL-4, and IL-5, which will increase B-cell proliferation. This progression was discovered on experiments on mice. Radiation will kill all the immune cells in the body, and it is a good method to test immune system variations. In this study, the control mouse was radiated and infused with RBCs from a sheep. This mouse did not mount an immune reaction. The second series of mice were radiated and had their immune systems reconstituted with bone marrow, so they were able to produce B cells. When given sheep RBCs, they did not mount an immune reaction. This means the B cells cannot respond effectively to the antigen. The third series of mice were radiated and their immune systems were reconstituted with cells from the thymus. When given sheep RBCs, they did not mount a response. The T cells alone could not respond effectively to the antigen. In the fourth series, the radiated mice were reconstituted with bone marrow and thymus cells. These mice could mount a response to the sheep's RBCs when given. Both the B cells and the T cells were needed for the response. This is T-dependent B-cell activation. Since the B-cell recirculates before it is activated, it frequently comes back to the lymphoid tissues on its journey. Once activated in the lymphoid tissue, the B-cell has three choices of what happens next. They can multiply and enter the primary follicles, forming a germinal center in the lymph node and remain there. This requires the help of BCL6 and not much help from IRF4. They can also become memory cells, which have the antibody embedded in their membranes. They circulate around, and when they find their cognate antigen, they mount a memory response. The third path is to become plasma cells. These cells form what is known as a primary focus from which they produce and release IgM antibodies into the system. This requires the help of IRF4 and BLIMP1. The activated B cell is now a long-lived cell in the body. This concludes the video on the T-cell dependent B-cell response for activation.